We are a bit behind our schedule. That means we have some things to cover until Thursday, so that we will have time to revise the contents of the report part for your preparation for the exam on Friday. For that reason, I will be a little bit in hurry today to cover these parts, finishing the sex differentiation a little bit, and then we are trying to finish this part of the male reproduction, dealing with the anatomical structure of the male reproductive system. We are trying to see the different cellular components of a testis, looking very close, look up to the testis, and then what are the different types of somatic support cells present in the testis, and what is their function, how the structure of the sperm looks like. We have already seen that one, but just for purpose of revision. At the time of puberty of the male, which type of hormones will be activated in the hypothalamus pituitary gonadal axis of the male reproduction. So to start with, the picture which you see there on the slide is showing the different sites of testosterone action. So we say that the Leydig cells are the Leydig cells in the testis are the ones which are producing the testosterone. This testosterone is a hormone which is acting on the various types of cells in the reproductive tract. And those cells which are forming the reproductive tract of the male are having the so-called androgen receptor, which is permitting the testosterone to act on those cells. Once testosterone is inside these reproductive cells, it will be in some of the reproductive organs or the reproductive parts, it will be changed to the so-called dihydrotestosterone. This transition or the change of testosterone to dihydrotestosterone is mediated by the enzyme called 5-alpha reductase. So testosterone, which is produced by the Leydig cells in the testis, will be transformed to dihydrotestosterone. Dihydrotestosterone is the same form of testosterone, but it is 4 times potent or active than the normal testosterone. So this dihydrotestosterone is responsible for the development of the different types of structures in the male reproductive. For example, the scrotum, the penis, and the prostate are the site points where we have higher activity of this 5-alpha reductase, changing testosterone to dihydrotestosterone. The other parts which are targets for testosterone are, which are, for example, the epididymis, the vas deferens, and the seminal vesicle. These are the site points of action of testosterone. However, in the other ones, testosterone will be captured by the cells, but will be changed to dihydrotestosterone so that the development of these organs takes place. That's why this picture shows how the different parts of the male reproductive tract are either they are dihydrotestosterone dependent or testosterone dependent only. Okay? So testosterone is a very important male sex hormone, which is not only important for the development of the different structures in the male reproductive tract, but they are also important for development of different phenotypic characteristics at puberty. The growth of facial, axillary, and pubic hair growth is dependent on that. The deepening of the voice is regulated by testosterone, and various types of structure and phenotypical characteristics which we see in a male at puberty is regulated by testosterone and dihydrotestosterone. To summarize what we have learned in the last 2 lectures. So the sex differentiation in the male is starting with XY chromosome. That means a male having XY chromosome, an individual having XY chromosome is a male, and this will lead to the presence of the SRY gene. We have seen that one. This is just a summary, the summary, so that you have every information in one picture. So the presence of SRY gene giving rise to the primordial gonad to develop into a testis. And the testis has 2 supporting cells. One is the Sertoli cells, the other ones are the Leydig cells. The Sertoli cells are the ones which are producing the anti-Mullerian hormone, or the Mullerian inhibitory substance, which is responsible for the regression of the Mullerian duct. When the Mullerian duct is regressed, the female reproductive development will be blocked, and the male side will proceed. So through the action of Leydig cells, testosterone will be produced in the fetal ovary, and this testosterone will be responsible for the Wolffian duct to be transformed into the different parts of the male reproductive system. That is the epididymis, the vas deferens, the seminal vesicles, and actually the ducts are derived from this Wolffian duct. So through the action of 5-alpha reductase in some of the cells of the reproductive tract, testosterone can be changed into dihydrotestosterone, which is leading to the development of penis, scrotum, and prostate. Very clean, very clear. You need to, if you know that one, you know a lot. Okay? So that is very summarized one. What about the female side? The female is differentiating in a different way. Because of the XX chromosome composition in the female, then we don't have any SRY gene. If there is no SRY gene, as I said in the previous lectures, the female development path is a default path, which is leading to the absence of SRY gene, leading to the differentiation of the primordial gonad to a fetal ovary. The fetal ovary is responsible for the Mullerian duct to be transformed into the uterus, the fallopian tube, and inner vagina, because of the absence of the Mullerian inhibitory substance, which was supposed to be normally produced by the Sertoli cells in the testis. Since the testis is not there, since only it is an ovary, this Mullerian inhibitory substance is not there, so the default program will be going so that the Mullerian duct will be transformed into uterus, fallopian tubes, inner vagina. So because of the absence of the Leydig cells, there is no testosterone. If there is no testosterone, the Wolffian duct will regress, and there will be no development of the male reproductive tract. So and then the development of the outer vagina and the female external genitalia will follow through the process. So this is the summary of both male and female sexual differentiation, which is regulated by the different types of things. So the one which I'm going to explain is just for your information. No need to keep it in mind, but so that you know that which kind of errors can happen during the time of spermatogenesis and oogenesis, especially at the stage of meiosis. During the process of meiosis, sometimes things can go wrong, and different types of sexual reproductive disorders can happen. One of the sexual development disorder is called the Klinefelter syndrome. Klinefelter syndrome is characterized by the presence of additional X chromosome in the male. Sorry. So this additional X chromosome in the male can be because of 2 reasons. One can be because of the maternal side, that the 2 X chromosomes will not separate, and an egg will be produced having X chromosome. Normally should have only 1 X chromosome. In the father side, can be possible that some of the sperms may have both XY as in their sperm genetic DNA. And if these individuals are mating with one Y, then this will lead to XXY individuals. These individuals are normally genetically male, but they are showing a lot of female characteristics. Their reproductive development, like the penis and other parts development, will be diminished. This is very simple to see here. That is an individual with XXY chromosomal combination. So this individual is having a more female characteristics, despite the fact that he is genetically a male individual. So it has a relatively prominent breast you see here. The penis is underdeveloped, and the pubic hair structure, which is more or less similar to the female. So these are some kind of errors which can happen during the time point of meiosis. This is just simply for your information. If you want to read more about these things, you can read more in Google or in other biological. So what is a Klinefelter syndrome? What does it make? And how the individuals are developing. This is for more reading. There are a lot of resources available. But for me, interesting thing is that sometimes things during the spermatogenesis or oogenesis can go wrong, which result in such kind of abnormalities. The other one is errors during the crossing over of X chromosomes. Under normal circumstances, so when crossing over happens during meiosis 1, the crossing over does not involve the SRY gene. So if you have here, if you have here SRY gene in the Y chromosomes, when crossing over happens, then you will have sometimes, under normal circumstances, this crossing over will not involve the SRY gene on the Y chromosome. However, sometimes this will involve and result into an X chromosome having SRY gene on it. So these individuals will have, for example, if they are XX individuals, they are genotypically they are female, but they can develop to a male phenotype because of the presence of SRY gene in one of the X chromosomes, because of error in X chromosome crossing over. So errors can happen in the separation of the two X chromosomes, or errors can happen at the time point of crossing over, as you can see here. This is a male phenotype. Such kind of infants have showing a very simple, a masculinized development of the penis because of the translocation of SRY gene on the X chromosome, which is normally not possible, but in rare cases it can happen, and that can lead to the excessive production of testosterone or androgen because of the action of SRY gene. Just for you to know that there is also sometimes errors happening during the crossing over, resulting such kind of phenotype. The other one is there is a so-called mutation in androgen receptor gene. Mutation in androgen receptor gene is leading to the inactivity of the androgen receptor, so that those individuals, they are not responding to testosterone. if androgen receptor gene is not active, so those tissues which are requiring for their development the androgen receptor, they will not be active. So because of this fact, individuals which have X, Y, males which have XY, they are genotypically male. But because of the fact that androgen receptor is not functional, they have a phenotype which is female. The picture which you see on the slide is showing an individual with XY chromosome. But because of the inactivity of the androgen receptor, this individual is showing a characteristics of female. Even the testis, which is present as in the nut body, is not descending out of the body, showing no signs of development of sexual organs like testis. The testis is in the body; it's not descending out of the body. The other one is they are phenotypically, as you can see, they are female. So as I said before, no need to worry about the different types of things, but you need to know that when you observe such kind of phenotypes or whatsoever in your day-to-day life, it can arise from different errors happening during the process of spermatogenesis and oogenesis. That is the message which I have. So we go now directly to the male reproductive system. So as you can see here, the male reproductive system is consisting of different parts, including here, for example, let me clean that one first. So, as you can see in the picture, the main parts of the male anatomical structure, the male reproductive anatomy structure, includes the testis. This is a site where sperm will be produced, and the produced sperm will be going to the epididymis, where it will be stored and matured a little bit, and then leading to the contractile activity of the epididymis is leading to the release of the sperm to the structure called the vas deferens. The vas deferens is the one which is responsible for the transport of the sperm from the site of production to the site of ejaculation. And this tube is a vas deferens tube is getting into the seminal vesicle, where it, under normal condition, we are looking at the one side of the male reproductive system. Most of the things in the male reproductive system are impaired. Testis, we have two testes. And the vas deferens is also two. The epididymis is two. So these are paired structures which are found in the right and left side. And they are coming together, these two parts of the male reproductive system are coming together in the prostate. In the prostate, the tubes, the vas deferens from the left and right side are merging together, and this is the site point of urine production, which will be just for you to show for your orientation purpose. It's not part of the reproductive organ, but this is a urinary system, is on the top of the reproductive system of the male, and the urethra, which is coming from the bladder for the urine transport, is joining the vas deferens, or the junction, the joining of the two vas deferens leads to the formation of the ejaculatory duct. So the ejaculatory duct will join the urethra in the prostate and going into the penis as urethra, as you can see here. So there are main parts of the reproductive tract, there are accessory organs. The accessory organs are including the seminal vesicle, the prostate gland, and the bulbouteral fluid. Normally, when we are talking about sperm, we are talking about only the sperm cells. However, when you see a male ejaculate, it is a fluid structure. It's not only the sperm, it has also a fluid part. So this sperm plus the seminal fluid, it's called seminal fluid, is the one which is making semen. Okay? So the seminal fluid in the male reproductive tract is produced by the seminal vesicle, prostate gland, and the bulbourethral gland. So the fluid which is contained in the semen is produced by the seminal vesicle, prostate gland, and this bulbourethral gland. So this seminal fluid has some advantage. Seminal fluid, which is making the semen, the sperm cells plus the semen, has advantage in providing nutrients to the sperm. Has several components which are important for the sperm to nurture itself during the process of after production to the process of ejaculation. So the sperm, the seminal fluid is also providing a kind of buffering role for sperm in the acidic environment of the vagina. Under normal condition, the vagina is very acidic environment, which is very toxic for the sperm, but the seminal fluid, which is containing the sperm, is providing a buffering or protection for the sperm from this acidic environment in the vagina. The other one is this nutrient or this fluid is providing containing chemicals which are important for movement or sperm motility. So when the sperm is produced initially, the sperm which is produced by the seminiferous tubules is not active to make fertilization of the egg. It has to be, matured and get maturity, and then should be capacitated. So maturity of the sperm is taking place normally after it is produced in the seminal phase, in the seminiferous tubules, and maturation of the sperm is achieved in the epididymis. And once passing through the whole parts of the male reproductive tract, after ejaculation, the sperm is stored in the vagina. During its travel or its tour from the site of ejaculation to the site of fertilization, is passing through the vagina, the uterus, and getting into the fallopian tube, where it meets with the egg. So the sperm is getting capacitated. So we will see that one next week on Wednesday when we are talking about fertilization process. Okay? So the sperm which is produced, which is ejaculated to the vagina, is not capacitated. It's not getting the capacity to fertilize, but capacitation happens through its travel through the vagina, the uterus, and the fallopian tube. So capacitation of the sperm is taking place in the female reproductive tract. Okay? So these are the function of the seminal fluid, just for you to have some idea what this specific fluid which is present in the semen is providing for the sperm. Looking into closer to the semen, the testis of a male, we will see that different structures. The first structure is the site of the sperm production. It's called seminiferous tubules. Sperm is produced in the seminiferous tubules. Between the seminiferous tubules, we have the so-called interstitial space or interstitial cells. These are the so-called Leydig cells which are producing testosterone. Okay? Testosterone production and sperm production is happening in different structures of the testis. And the sperm which is produced by the seminiferous tubules is traveling to the epididymis, which have two parts. So seminiferous tubules are the sites of sperm production. Interstitial cells, these are the so-called Leydig cells, are responsible for testosterone production. Testosterone production. And the sperm which is produced by the seminiferous tubules is going to the epididymis, where I say that it will be stored and maturation takes place. Epididymis, which has two parts, the head part and the tail part. The sperm which is produced by the seminiferous tubules is not something which is fluidic. So the transport of the sperm from the site point of production to through the epididymis to the vas deferens is maintained by the contraction and release of the smooth muscles of the epididymis. So epididymis is important for the storage and maturation, and as well as for transport of sperm from the site point of production to the vas deferens. Okay? So when you look into a testis, it is normally found outside of the body. There is one reason for that. The sperm, the testis is under normal condition, is found like this, like two structures which are separated by, that is the scrotum, that is the body part, and that is the testis on both sides. So for sperm production to happen, the optimal environment in the testis should be a lower temperature, which is normally 2 degrees Celsius lower than the body temperature. This can be attained because of the fact that this testis is hanging out of the body, so that there will be circulation of air. This circulation of air will allow the testis to have relatively lower body temperature, so that spermatogenesis can happen. So this is one way that the sperm is hanging out of the body to attain lower temperature. The other way is exchange of heat. Mediated by blood flow through arteries and veins. So exchange of heat is also mediated by arteries and veins, which are also involving or which are also contributing to a relatively lower temperature in the testis so that spermatogenesis can optimally happen in the testis for that purpose. So these are the two reasons why we have, so as I said, the hanging of the scrotum outside of the body is allowing the air circulation to happen so that testicular temperature is lower than the body temperature by 2 degrees Celsius mostly. And the arteries and the veins which are coming to the testis and going out of the testis are exchanging heat so that this optimum temperature can be maintained. If we look into we have already seen that one. If we look into the cross-section of the seminiferous tubules where sperm is produced in the testis, we will find different structures. This is electronic microscopy showing that this is seminiferous tubules of the adult testis. Between the seminiferous tubules is these interstitial cells. These are called the Leydig cells, where the site of production of testosterone. And in the seminiferous tubules, we have these so-called Sertoli cells. The Sertoli cells have two functions here. One is they are providing nutrients for the developing sperm, and the other one is that they are providing a tissue barrier so that there will be no blood coming into the lumen. Okay? So they are located around the seminiferous tubules, and they are used as a kind of barrier so that there will be no blood coming into the seminiferous tubules. So the spermatogonial cells here, what we see here at the edge, they are traveling through this process of spermatogenesis to form the spermatids, which you see here. These spermatids will get matured and released into the lumen, having a tail, what you see here, the different structures of the sperm. Okay, this is just structural parts of the seminiferous tubules. We have already seen that one. There is one thing which I want to mention here, that compared to the female, we have already said that mitosis of the spermatogonial stem cells in the male is starting at puberty, going throughout the life. I think everybody will remember that. In case of females, mitosis is already completed during the fetal period, and a baby girl is born with primary spermatocyte, primary oocyte. For this continuous process to happen throughout the life of a male individual, there is this process which is called that one of the clones of spermatogonial stem cells will be used as a feedback loop to be used as an input for the next round of mitosis. So all which are undergoing mitosis are not going to primary spermatocytes. Some of them are going back to be used as an input. So that is one of the characteristics which is not present in the female. This is the reason why a male is able to produce sperm throughout the life. However, in the female, we say that around 400,000 primary spermatocytes are present. Primary oocytes are present at the time of birth, and that is the reserve what a female has to be utilized when the female is at the age of puberty through its menstrual cycle. Yeah, so that is the difference. The male can do that spermatogenesis, having this feedback loop that one of the clones can be used as an input for the other round of mitosis, which will be so that the whole spermatogenesis will be a continuous process throughout the life. There is nothing special on this one. We have already seen that one. So the head, the sperm has three structures. We have already seen that, but just for you to summarize. We have the head, we have that is the head of the sperm. It has here an acrosome, which is having different types of enzymes which are important for the penetration of the sperm through the zona pellucida of the egg. And the head has the DNA, which is the genetic information is here packed into the head of that one. The mid piece is the next phase, the next part, which is having the mitochondria of the sperm. The mitochondria of the sperm is providing energy for sperm motility. The tail part is composed of several microtubules. That's the tail. These microtubules are a kind of flagellar structure. They are important for the forward movement of the sperm. Okay? So these three parts, we have already seen that one. So the head of the sperm is containing the acrosome and the nucleus. The mid piece is containing the mitochondria, and the tail part is once the male is reaching to puberty, in that case, the so-called hypothalamus pituitary gonadal axis will be activated. That is something which is happening both in male and female, but we will see today how during at puberty, how this the hypothalamus pituitary gonadal axis. So so in the brain, the so-called hypothalamus is a place for the production of the hormone called GnRH. That is gonadotropin releasing hormone. Gonadotropin So the gonadotropin releasing hormone is released from the hypothalamus. That's why we said HGC means stands for hypothalamus. This release of the GnRH is acting on the pituitary, the anterior pituitary. Pituitary to release two types of hormones here. One is FSH, follicle stimulating hormone, and LH is luteinizing hormone. These both hormones are present, those including that one, present in the female reproductive tract too. Okay, in the female reproductive. Even the name itself of these two hormones is coming from their function during puberty. As you can see here, follicle stimulating hormone is, this hormone is responsible for the recruitment of follicles in the ovary. The LH is, the luteinizing hormone, is responsible for the luteinization of the granulosa cells in the follicle for the formation of the corpus luteum, and this one is responsible for the LH surge is responsible for ovulation. But now we are talking about only the male side. We will talk on the female side on Wednesday. So these hormones, they are acting on the testis. In the testis, we have two types of cells, two types of supporting cells. These ones are the Sertoli cells and the Leydig cells. At puberty, the FSH is acting on the Sertoli cells, the LH is acting on the Leydig cells. The Sertoli cells, through the action of FSH, they are producing the so-called inhibin. It's a protein hormone, which is important for the negative feedback, regulation of FSH release. So hormonal release from the GnRH, from the hypothalamus, anterior pituitary, that is the pituitary, and that is the gonad. From the anterior, so the secretion of hormones is a pulsatile process which is regulated by the negative feedback loop of some hormones produced in the ovary, in the testis. In this case, the Leydig cells, through the action of LH, they are producing testosterone. So this testosterone, which is produced by the Leydig cells, can be used in two ways. One is providing support for the Sertoli cells, so that Sertoli cells can be involved in the production of inhibin, and also they are involved in the process of spermatogenesis, as I showed you how they are localized in the seminiferous tubules. They are important for spermatogenesis. So testosterone can be locally used by Sertoli cells to support the process of its contribution to spermatogenesis, and externally testosterone can be used for various reproductive purposes. Outside of the testis. So at puberty, when we are talking that a certain individual is at puberty, sexually matured, that means this axis is active. Before that, this axis is not active. So once this axis is active, the hypothalamus in the brain is producing GnRH, which acts on the anterior pituitary, so that this anterior pituitary is producing two types of hormones, the FSH and LH. The FSH is acting on the Sertoli cells of the testis, and these Sertoli cells are producing inhibin for negative feedback of the FSH and involved in spermatogenesis. And the Leydig cells, through the action of LH, are producing testosterone, which is locally used by Sertoli cells to promote spermatogenesis and also externally to produce, and also giving the other side of the pituitary hormone is regulated by the action of testosterone, is acting on inhibiting the synthesis of LH, and also directly on the synthesis of the GnRH. So testosterone is regulating most of the processes by having a negative feedback loop directly on the LH, indirectly through the action of its effect on the GnRH. So inhibin is responsible for the negative feedback loop with FSH. That is the system which you need to just emphasize, that at puberty, this axis is active. At the time points of maturity or puberty, the hypothalamus is involved in generating GnRH. GnRH is acting on pituitary. The pituitary are producing two types of hormones. The FSH is acting on testis, Sertoli cells. The LH is acting on Leydig cells. The Leydig cells are producing testosterone, which can be locally utilized in the testis for promoting the activity of the Sertoli cells so that they can support spermatogenesis, or it can be involved in other types of functions in the reproductive tract. As I said before, at puberty, testosterone and dihydrotestosterone regulating various types of phenotypes. The growth of hairs, the deepening of the voice, and all different types of phenotypic characteristics what we see in a male at puberty is regulated by this testosterone. So they are also involved in regulating the synthesis. So a continuous production of these hormones will not be optimum. That's why this regulatory effect should be there so that the cycle will continue. Okay? So this is just summarizing what the hormonal control of the testicular function looks like. As I said before, the two supporting cells which are important in the testis are, one of them is the Leydig cells, which are lying between the two seminiferous tubules. They are synthesizing and releasing testosterone, as I showed you here. And in the testis, we have seen that where sperm production is happening in the seminiferous tubules and testosterone production is happening in the Leydig cells, showing that sperm and hormone production are in different compartments in the testis. So as I just explained, the Leydig cells are stimulated by LH to produce testosterone, and testosterone increases the activity of Sertoli cells for their contribution to spermatogenesis. The other important cell types, or the other important supporting cells in the testis, are the Sertoli cells, which are forming a tight junction to have a blood testis barrier, and also they are supporting the developing germ cell, the sperms, by providing nutrition and also providing physical protection. And they are secreting also some kind of fluids which are containing androgen binding protein, and they are responding to testosterone FSH as a paracrine agents to stimulate sperm proliferation and also differentiation of the germ cells. So as I said before, Sertoli cells are producing inhibin, which is a negative feedback regulator of FSH, as I showed in the testis, also indicated in that one. They are also involved in phagocytosis of dead sperms or defective sperms. So dead sperms and defective sperms will be phagocytized by Sertoli cells in the testis. In fetal life, we have already seen in the fetal testis, the Sertoli cells are responsible for the production of anti-Mullerian hormone, which is leading to the regression of the Mullerian duct, so that the female reproductive tract will not be happening. I think that is all. We have already tried to cover how the reproductive tract in the male looks like. What are the different components of the testis? We have seen already, yeah? So the epididymis, the seminiferous tubules, the vas deferens, and all others. The two supporting somatic cells, we have seen that Sertoli cells, and also the Leydig cells are, how their function is. We have already seen what the sperm structure looks like, the head, the midpiece, and the tail of the sperm, what they're for. So what is the hypothalamus pituitary gonadal axis looks like at puberty in the male. I think that is all is covered in this one. So, okay? So if you have any questions, just bring in the next section or just write me an email so that we are in a position to cover most of the things which we have planned to cover, so that we will have time on Friday to revise for the exam. Okay?
Thank you.
Yeah, welcome.